Good morning, dear students. We are with the topic of second year microbiology. Some basic aspects regarding enzymes. फर्स्ट इयर मध्ये आपण जे बेसिक जो कॉन्सेप्ट शिकलेलो होतो त्याच्यापेक्षा आता ॲडव्हान्स कॉन्सेप्ट मध्ये आपल्याला जायचं आहे. एन्झाइम्स, प्रोटीन्स, कार्बोहायड्रेट्स हा पार्ट आपल्याला सेकंड सेमिस्टरला वगैरे होतो. बट नाव वी आर गोइंग टू गो इन अ लिटल बिट डीपर. What are enzymes? What are the applications of enzymes? How enzymes actually work? What are the mechanisms by which the working of an enzyme can be easily understood? या गोष्टी आपल्याला आता सेकंड इयर मध्ये बघायच्या. So let us begin with our lecture. So some basic aspects of enzymes. Two fundamental conditions of life, that is self-replications and able to catalyze chemical reaction. Enzymes are biocatalysts that catalyze life. Enzymes are central to every biochemical process. The study of enzymes has an immense practical importance. Study of enzyme is called as enzymology. So the basic concepts of enzymes are these are biological catalysts. These are said to be biological catalysts, and these catalysts are helpful for conducting various different biochemical processes. Every enzyme has its own activity, and this activity will be helpful for that enzyme to work on a specific substrate, degrade the substrate into product, and this product will be utilized in many biochemical reactions. Now, when we are defining in terms of catalyst, so what is a catalyst? A catalyst by its mere presence affects the rate of reaction without undergoing any chemical change itself. This is called as a primary or a basic definition of the process of catalyst. So biological catalyst, why it is called as biological catalyst? Because enzymes are being produced by bacteria, by microorganisms, and these enzymes are helpful for conducting various different metabolic reactions or metabolic actions. So once they participate in these chemical reactions, the amount of catalysis that takes place is much more faster, is much more safe, and the exact product from that particular substrate after degradation will be there. Again, the study, since it is logy means study, yesterday only we have seen this part. So this study of enzymes is called as enzymology, and in the coming lecture we are going to see what are the applications of this. Let us first understand what is the history of enzymes, from where the enzymes actually have started. Biological catalyst was first recognized and described in the 1700 digestion of meat by gastric secretions. In the 1800, the conversion of starch to sugar by saliva and various plants extract was studied. In 1850, Louis Pasteur introduced the term ferments, fermentation of sugar into alcohol by yeast. Emil Fischer discovered in 1894 the glycolytic enzymes which distinguish between the stereochemistry sugar led to the formation of Locke-King hypothesis. In 1897, Edwin Boucher discovered the yeast extract for fermentation of sugar and alcohol. Means, as we are going to study the enzymes, these enzymes have been studied a lot by various different scientists. आज आपण एन्झाइम स्टडी करतो याचा अर्थ असं नाही की ते एन्झाइम आजच्या आजच प्रोड्यूस झाले. इट इज बीन produced by bacteria or produced by the living cells from a long period of time, and all these scientists have been working on different aspects of enzymes, and they have been producing enzymes a lot from various different sources. काही ना असं माहिती पडलं की from digestion of meat the enzymes are there. In your saliva and various plant extract there are enzymes that have been present. Pasteur has shown in terms of fermentation that conversion of sugars into alcohols by yeast. This was one of the best examples in microbiology. Fischer discovered the glycolytic pathway and the enzyme Locke-King hypothesis as well. And Boucher, yeast extract from fermented sugar to alcohol again was proved by this particular scientist. Frederick Cohen later gave the name enzyme to the molecules related by Boucher. In 1926, the isolation and crystallization of urease by James Murray. हा एक खूप टर्निंग पॉइंट होता या पर्टिक्युलर मेथडचा किंवा या पर्टिक्युलर टॉपिकचा, बिकॉज द फर्स्ट स्ट्रक्चर ऑफ एन्झाइम, आता कॉम्पिटिटिव्ह एक्झाम मध्ये पण विचारला जातो, विच इज द एन्झाइम which is among the following enzyme the exact crystallographic structure was known for the first time, that is the enzyme urease नावाचा एन्झाइम, आणि कोणी सांगितलं? जेम्स समनरनी ते शोधलेलं होतं. He postulated that enzymes, all enzymes are proteins. In 1930, protein's nature of enzymes is widely accepted from John Northcott's and Moses Crude's crystallized pepsin and other digestive enzymes found them also be protein. In 63, the first amino acid sequence of the enzymes, that is bovine pancreatic deoxynuclease A, was reported, and X-ray structure of enzymes, the hen egg white lysozyme, was elucidated in 1965. मग हे पर्टिक्युलर सायंटिस्ट लोक शोधत गेले, अजून खोलामध्ये गेले की एन्झाईम्स कसे असतात, काय असतात. तर अल्टिमेटली द ऍक्टिव्हिटी ऑफ एन्झाईम जेव्हा बघितली गेली तर ऑल एन्झाईम्स आर सेड टू बी प्रोटीन्स. एक गोष्ट लक्षात घ्यायचं, ऑल एन्झाईम्स आर प्रोटीन्स बट ऑल प्रोटीन्स आर नॉट एन्झाईम्स. कारण का की एन्झाईम्स, अजून एक एन्झाईमला एक स्पेसिफिक शब्द आहे, जेव्हा एन्झाईम आपण ते प्रोटीन केव्हा म्हणू शकतो, व्हेन दॅट प्रोटीन इज कॉल्ड एज अ फंक्शनल प्रोटीन. करेक्ट. When it is a functional protein, it is called as an enzyme. But when it is a structural protein, it is not at all called as an enzyme. म्हणूनच ही टर्म फार महत्त्वाची आहे की ऑल एन्झाईम्स आर प्रोटीन्स. सगळे एन्झाईम्स काय आहेत, प्रोटीन्स आहेत, बट ऑल प्रोटीन्स आर नॉट एन्झाईम्स. बेअर इन माइंड, हा गोष्ट लक्षात ठेवण्यासारखी. सगळे एन्झाईम्स हे प्रोटीन्स आहेत, पण सगळे प्रोटीन्स हे एन्झाईम नाही. देयर आर फंक्शनल प्रोटीन्स which are enzymes. Structural proteins are not at all called as enzymes. Correct? Correct. Some basic definitions. Defined as biocatalysts synthesized by living cells. Enzymes are proteins that speed up the rate of chemical reactions in a living organisms. An enzyme acts as a catalyst for specific biochemical reaction, converting a specific set of reactants, that is substrate, into specific products. Substrate, enzymes are products. So this is how the reaction of an enzyme would take place, or ultimately the basic definition of enzyme can be worked out. Biocatalyst, I is a biocatalyst, and enzyme ची फोड कशी केलेली त्यांनी, an enzyme. Proteins, chemical ची reaction, पूर्ण chemical reaction speed up म्हणतात. Acts as a catalyst for reaction, converting specific reactants. म्हणजे काय करतं, सबस्ट्रेट वरती एन्झाईम ऍड करणार आणि याच्यापासून काय डेव्हलप होणार, the product will be possible. Means possible reaction or conversion of substrate into product is catalyzed by the enzyme. All the enzymes are protein except ribozymes, and number of enzymes are obtained in crystalline form. It acts on substrate to change it into products. हे सगळ्यात महत्त्वाचा पार्ट आहे की तो स्पेसिफिक एन्झाईम असणार आहे, स्पेसिफिक प्रोडक्ट वरतीच तो जाऊन रिॲक्ट करणार आहे, इकडे तिकडे आजूबाजूला जाणार नाही. Their molecule weight ranges from few thousands to million Daltons. Enzymes are far more efficient compared to non-enzyme catalysts. Enzymes are not consumed in overall reactions. Enzyme accelerate the rate of reactions but does not alter any changes. हेच मेन कॅटलिस्टचा हा सगळ्यात महत्त्वाची भूमिका कॅटलिस्टची हीच असते की the catalyst is helpful for development of the reaction, one, for fastening or speed up the reaction, second, for converting that substrate into quality product, and the product will be utilized for other biochemical reactions. हा महत्त्वाचे 3-4 पॉइंट जे आहेत हे या पर्टिक्युलर केमिकल रिएक्शन्सना एन्झाईम्स कशी मदत करतात, that is well been defined. Some enzyme does not require chemical groups for activity other than that of amino acid residues. Other enzymes requires additional chemical components such as cofactor or coenzymes. आता नवीन कॉन्सेप्ट आहे थोडासा cofactor आणि coenzymes की these cofactor and coenzymes are nothing but they are helpful for generating or they are helpful for making that substrate into product B. म्हणजे एक एन्झाईम आहे, तो एन्झाईम ऍड करतोय त्या पर्टिक्युलर सबस्ट्रेटवर and it is converting that substrate into product B. पण हा प्रोडक्ट जेव्हा तयार होतोय, ह्या प्रोडक्ट मला हे जे काही कन्वर्जन आहे या कन्वर्जनला त्या एन्झाईमला सपोर्ट करण्यासाठी the most important two components are essential, that is cofactors and coenzymes. हे cofactors and coenzymes हे स्पेसिफिकली त्या एन्झाईम वरती डिपेंडेंट आहे की कोणचं एन्झाईम आहे, आणि हे खूप छोट्या प्रमाणात, खूप लहान प्रमाणामध्ये लागतात, बट they are helpful for converting the product, converting the substrate into product. Some enzyme proteins are modified covalently by phosphorylation, glycosylation and other processes. They are colloidal in nature, heat liable and water soluble. They can be precipitated by protein precipitation reaction, ammonium sulfate or trichloroacetic acid. आता प्रोटीन आहे, आपण प्रोटीन मागच्या वेळेस सेम टूला शिकलेलो आहोत. प्रोटीन्स आर नथिंग बट दीज आर स्ट्रक्चर और चेन्स ऑफ अमिनो एसिड्स, करेक्ट, बाउंड ईच अदर इन प्रायमरी, सेकंडरी, टर्शरी, क्वाटर्नरी असे 4 प्रकारचे स्ट्रक्चर्स आपण बघितले होते, त्यांचं बॉन्डिंग आपण बघितलं होतं मागच्या सेमिस्टरला. आता हे जे काय, दीज आर ऑल प्रोटीन्स इन नेचर, सो दे आर हीट लेबाइल, दे आर व्हेरी मच सेन्सिटिव्ह, दे आर स्पेसिफिक. ह्या ज्या प्रॉपर्टी प्रोटीनला ऍप्लिकेबल आहेत, द सेम प्रॉपर्टी इज ऍप्लिकेबल टू दिस पर्टिक्युलर काइंड ऑफ एन्झाईम. सो त्या एन्झाईम्सही ते वर्कआउट ते करू शकतात. Structure of enzymes. Functional enzymes is a hollow enzyme. Functional, लक्षात घ्यायचं. Hollow enzyme with protein, apoprotein and non-protein coenzyme. लक्षात घ्या. Prosthetic group are coenzymes or metal ions that are very tightly or evenly covalent bound to the enzyme. हे पहा. हे enzymes hollow enzyme आहे. या hollow enzyme चा हा apoenzyme त्याला cofactor अटॅच झाला, substrate अटॅच झाला. Cofactor जेव्हा apoenzyme ला attach होणार आहे then it is called as hollow enzyme, and this coenzyme, cofactor and hollow enzyme will be helpful for the substrate to react into it. Single polypeptide monomeric enzymes, that is trypsin ribonuclease, polypeptide change, oligomeric enzymes, lactate dehydrogenase, hexokinase enzymes चे प्रकार आहे. Multi enzymes processing specific site to catalyze different fatty acid synthase, carbonyl phosphate synthetase 2, pyruvate dehydrogenase, prostaglandin synthetase, etc. The complex becomes inactive when it is fractioned into smaller units, each being individual enzyme. आता enzymes जसं म्हटलं आपण की enzyme काय करतोय, enzyme substrate वरती react करतोय and convert it into product P. पण सगळे enzyme एकाच सारख्या substrate वर react करतात का? No. There is specificity between an enzyme and its substrate as well. जशी substrate ची specificity आहे तशी enzyme ची पण specificity ही आहेच. So this enzyme will be helpful for reacting with the substrate and convert it into product P. Cofactors. So the term cofactor is used collectively. Term include the coenzymes and metal ions. Coenzymes is organic cofactor. They help in either maintaining or producing both active structural conformation of enzyme. म्हणजे enzyme ला structurally conformation नीट व्यवस्थित राहावं याच्यासाठी कोण काम करताय तर काम करतात cofactors आणि coenzymes. They help in the formation of enzyme substrate complex. Cofactors helps in making structural changes in substrate molecules. Cofactor accept or donate electrons activating on functional as nucleophiles and formation of tertiary complex as well. म्हणजे हे काय करतात cofactors? These cofactors help the formation of enzyme substrate complex. आता आपण सरळ एक रिएक्शन बघितली होती की there is one enzyme and there is one substrate. Correct? This enzyme will act on that substrate and it convert it into product. Fine. पण हे enzyme substrate complex जे तयार होणार आहे that is called as ES complex. Enzyme for E, S for substrate. Enzyme substrate complex. So this enzyme substrate complex, the stability of enzyme substrate complex is said to be very much important. आणि त्या बेसिस वरतीच the conversion of that substrate into product P is said to be very much essential. Correct? See some inorganic ions that serve as cofactors for enzymes: copper, cytochrome oxidase, Fe2 plus Fe3 plus, cytochrome oxidase catalase peroxidase, K plus pyruvate kinase, Mg2 plus hexokinase, Mn2 plus arginase, MO dehydrogenase, Ni2 plus udase, Sc glutathione peroxidase, and zinc, there is carbonic acid. So these are all enzymes and these are the metal ions that are helpful. Now cofactors are said to be nothing but metal ions, and these metal ions are helpful for reacting along with that enzyme and convert it into product. Now coenzymes, the non-protein organic lower molecular substance associated with the enzyme is called as coenzymes. A coenzyme or metal ion that is very tightly bound to an enzyme forming prosthetic group. Coenzymes are second substrate, affinity with the enzyme compatible with another substrate. Participate in various reaction, oxidation, group transfer, so and so. म्हणजे coenzymes काय? Coenzymes is the non-protein moiety, but it is helpful for attaching that enzyme to its substrate in a specific manner. See this. Coenzymes काय काय वेगवेगळे आहेत, त्याचा ग्रुप कोणते आहेत आणि त्याचा डायटरी प्रोसेस एनिमल्स कशामध्ये आहे. Correct? So we'll stop here for today. This is the very basic part. आजच्या पार्ट मध्ये आपण काय बघितलंय? तर आजच्या पार्ट मध्ये आपण बघितलंय what are enzymes, what are the basic definitions of enzymes, then what are coenzymes, what are cofactors, what the reactions of substrate takes place, or what in the enzyme reaction takes place. In the next lecture we try to classify that enzyme into different classes. Once they have been classified, it will be more better for us to understand how the actually enzymes work. Correct? Sure. Thank you.